I'm going to hazard a guess that all of you at some point have been to Wegmans or you've been to McDonald's and you've gotten yourself a drink from one of these big Coca-Cola fusion machines. The ones with the touchscreens where you get to choose your favorite flavor, your favorite soda, any add-ins that you might want, and it'll just dispense it right into your cup. And perhaps you didn't think about where all that soda was coming from. If you've got 50 different varieties to choose from, there's not a container that contains 50 different volumes of soda that are all just getting dispensed and eventually emptied into your cup. So there has to be some mechanism by which we have some concentrated soda syrup that gets mixed with carbonated water, and that concentrate ends up turning into the drink that you had ordered. That process of taking a concentrated syrup and mixing it with bubbly carbonated water to make a drinkable solution that's not super, super sweet or very bitter, depending on your flavor, is actually what's happening with dilutions. We're going to be using this concept of dilution this week and also later in the semester. But to really understand what's going on, first we have to describe how we understand the concentration of a solution in the first place, right? Because we want to be able to describe how much of a particular substance is present dissolved in a solution at different stages of an experiment. One way to do this, and one way that you likely learned in chemistry, is using the concept of molarity. Molarity describes the number of moles of a solute dissolved into 1 liter of a solution. And a mole, remember, is 6.02 times 10 to the 23rd molecules of a particular substance. For example, a 2-molar solution of sodium iodide would be calculated this way. Very simply, we're going to take a volume of sodium iodide that represents 2 moles of that chemical, and we'll dissolve it to water, which is our solvent, to a final volume of 1 liter. Now we need to figure out how much does 2 moles of sodium iodide weigh. And the way of calculating that is to figure out what the formula weight is, because 1 mole is the formula weight of a molecule in grams. So the formula weight of sodium is 23 grams per mole, and the formula weight of iodine is 227 grams per mole. So when they combine, we end up with a total mass of 150 grams per mole. That means 1 mole of sodium iodide weighed out and put into a dish is 150 grams. In our example, we want to have 2 moles, which means that we're going to have to weigh out a mass of 300 grams of sodium iodide and then dissolve that in water until it reaches a final volume of 1 liter. That would be a 2-molar solution. And this is a great way of calculating concentration for very simple molecules. Because if I was to make a 2-molar solution of sodium iodide, and you were in Los Angeles at a neighboring lab, and you wanted to make the same solution to do the experiment I was working on, you could follow the same recipe and you'd end up with exactly the same solution. So molecules like sugar, a monosaccharide, have a pretty standard molecular weight, and we would know that glucose has a formula weight of 180 grams per mole. So a 1 molar solution of sugar would be 180 grams dissolved in 1 liter of solution. Sucrose is a disaccharide. Sucrose is table sugar. It's what you put in your coffee. And that has a formula weight of 342 grams per mole. How much sugar does soda have in it? We're going to go back to our example of the Coke fusion machine. 1 liter of Coke has 106 grams of sugar. I'm going to simplify a little bit because the sugar in Coke is not strictly sucrose. It's fructose and other sugars. But it's pretty close for our experiment here. And we can calculate then what the molarity of sugar is in that 1 liter of Coke. We know from our example that Coke has 106 grams and that the molarity of sucrose is 342 grams per mole. That means that every liter of Coke has a concentration of 0.31 molar sugar. That's a lot of sugar, but we could calculate that pretty easily because we knew the formula weight and we knew a volume. This doesn't work quite as well if we're looking at big, complicated molecules for which our formula weight might be difficult or impossible to figure out. So we can think about starch, which is a molecule which we cannot determine the molecular weight. Starch is a polysaccharide made up of lots of glucose subunits bound to one another. Because we don't know how many glucose subunits there are for every molecule of starch, it's effectively impossible to come up with a molecular weight of a large polymer. So how do we go about making a standardized solution if we don't know the absolute molecular weight? And the answer is to use percentage instead. So let's use starch as our example here, and I'll describe how to make a solution based on the concept of percentage instead of molarity. We know that starch has variable lengths, right? We have a polymer that could be 10 glucose subunits or as many as 100,000 subunits. This is also useful when we're considering complex chemicals or mixtures of chemicals that might be made up of lots of different components. Microbiological media is a great example because many medias are just made up by dissolving an extract from something complex like a soybean. And we don't know exactly what the percentage of sugars or lipids or amino acids might be. So in these cases where we don't know the molecular weight, we can simply use the percentage of a solution, meaning a 1% solution represents 1 gram of a solute, some complex unknown molecular weight solute, dissolved in water to a final volume of 100 milliliters. And the basic assumption is that water has a volume of 1 gram per milliliter. So 1 gram is approximately equivalent to 1% of a 100 milliliter volume of a solution. It's not always going to be perfect, but our weight to volume calculations here will standardize our creation of a solution. So again, a 1% solution represents 1 gram of a solute dissolved to a total volume of 100 milliliters. As an example, a 6% solution of sodium chloride, regular table salt, would be 6 grams of sodium chloride plus a volume of water that would give us a final volume of 100 milliliters. I don't say that we're going to add 94 milliliters of water because I can't guarantee that in every case, 6 grams of our solute is going to represent a full 6 milliliter volume after being dissolved. So now we know that we can use molarity and we can use percentage as a means of describing how concentrated any given solution is that we are working with. Now we want to start creating dilutions by taking very concentrated samples of a solution and making it thinner or weaker by adding additional solvent to it. That's the general definition of a dilution. And it is often necessary for laboratory work because we'll prepare stock chemicals in a highly concentrated means because it's easy to store, and then when we use it, we would dilute it or make it weaker by adding some more solvent for a working volume that we might need to use. And it also ends up being a function of biological processes. Think about diffusion. When you are moving molecules from high concentration to low concentration, what you are in effect doing is you're creating a dilution. We're going to do a number of calculations here. Please don't make these very hard in your mind. They end up being very, very easy, especially because we're just going to use a single formula for all of our dilution calculations this semester. That formula is C1V1 equals C2V2, and it's just a simple ratiometric comparison of a before and after situation. So we're always going to be looking at a situation in which we have a before, where we've got a solution that has a concentration that is usually very high, and then we take a small sample of this high concentration solute, we move it into a new flask, we add a little bit of solvent to that, which results in a lower concentration. This would be your concentrated soda syrup, and then this is the soda that's drinkable after adding your carbonated water. Let's define these variables for you, because we want to be able to know how the concentration of a solute changes in a before and after situation, either as an experiment that we're doing ourselves, or, as we're going to see in this lab, as we look at high concentration molecules moving from inside of a cell to outside of the cell, which is effectively a dilution. So in our formula, C stands for concentration, which could be in molarity or percentage. And V represents volume, either our starting volume or our finishing volume. The subscripts, 1, represent our starting solution, or our concentrate that we're starting with, and the subscript 2 represents what we want to have after our dilution. And the good news is that as we're calculating these through practice problems or by looking at cellular dilutions, you're only ever going to need to solve for one of these variables at a time. Let's go through a walkthrough example of what you might see on a quiz or on a final exam. This is a fairly standard scenario that you might see in a laboratory. You have 250 milliliters of a 10% stock solution of sodium hydroxide. How much of this do you need to make 500 milliliters of a more dilute solution at a concentration of 2%? To start this problem, we can start assigning some of these values to the variables in our C1V1 equals C2V2 equation. And I usually like to draw a picture just to make sure that I am on the right page. So here's our scenario again. Here's our flask that has 250 milliliters, and the substance inside is 10% solution of sodium hydroxide. And I'm ultimately trying to get a total volume of 500 milliliters in another container. And this is going to be more dilute. It's going to be weaker, it's going to be thinner, and it'll eventually have a concentration of 2%. We're being asked how much of the blue liquid from flask 1 do I need to put into flask 2 in order to make that happen. Always start with the end in mind. So start with what you are trying to make, because that's going to make it easy. It can be very easy now to fill in all the variables and say, I've got nothing to solve for. So we are looking for a 2% solution. So let's assign 2% to C2, which is our final concentration. And our final volume we're looking to make is 500 milliliters. We are starting with, on the other side, a solution that has a concentration of 10%. And before you get ahead of yourselves, we are not going to say that V1 is 250 milliliters, even though that's the volume that we have in our flask. The reason being is we're not going to use all 250 milliliters. The question is, what portion of that 250 mils do we need to use? So in this question, V1 is currently an unknown. If we plug all these in, we end up with C1V1 is 10% times some unknown volume, which is equal to our concentration of our final product, 2%, times a volume of 500 milliliters. We can simplify this a little bit by getting rid of the units, because we know that we're looking for a volume, and that volume will be in milliliters. So we can simplify and just say 10 times V1 equals 2 times 500, and then just go through and continue simplifying. 10 times V1 is 1,000, V1 is 100 milliliters. So in practice, you would use a pipetter, and you would transfer 100 milliliters of our solution from the flask on the left over to the right-hand side. But we're not done. That's not the dilution yet. That's just saying this is the concentrate that we need to add to our flask, and then we're going to have to dilute it by adding water until we reach that 500 milliliter volume. So now we would do that, and our takeaway is a flask with 500 milliliters of solution, and the final concentration here is 2%. We've just taken our concentrated soda extract, transferred it over into our cup by adding a lot of carbonated water to it so that it is now drinkable. That was a pretty reasonable example of how you would make a solution using flasks and pipetters in the lab. But what if this change was happening over time? What if, instead, we started off with a flask that had about 500 milliliters of water in it, and we added a small cell with a selectively permeable membrane that had inside of it very concentrated salt. We could use sodium hydroxide or something else. Over time, those ions, if they have a means of getting across the membrane, are going to start diffusing out of our cell because they're moving from high concentration to low concentration. And if that was happening, then we could start asking questions about what factors might influence the rate at which this diffusion was occurring. How could we increase the rate at which our ions were moving from inside the cell to outside of the cell? That will be the focus of this week's lab. So remember that when you come, everybody needs to bring your computers. You should also bring your phylogeny and R assignments to turn in for grading. Quiz 3 starts on Monday morning, and it's going to be based on experimental design, diffusion, and some dilutions, meaning that you're going to have to be able to carry out the calculations in the formula that you just learned in this PowerPoint. So make sure that you bring a calculator. And don't forget to follow this up with video number 3 on how to use linear regressions to calculate rates.